Hello and welcome. Welcome to the UQ Art Museum for this inaugural Leonardo ISAST LASER Talk in Australia. I'd like to begin by acknowledging the traditional owners and their custodianship of the lands on which we meet, the Turrbal Yagara people. We pay our respects to their ancestors and their descendants who continue cultural and spiritual connections to country. My name is Grayson Cooke, and with my great friend and colleague Elizabeth Stevens, I'm overjoyed, excited, very pleased to welcome you to what is the first in a series of talks that we'll be convening with the support of Leonardo and the International Society for the Arts, Sciences and Technology. So you're here at a laser talk. LASER is an acronym. It might be a metaphor. Maybe these talks are about highly directed, attentional, focused discussion getting cut through, but in fact it is an acronym. Leonardo Art Science Evening Rendezvous. Tonight's talk is part of an international program of gatherings that bring artists, scientists, other scholars and the public together for presentations, performances and conversations. The mission of LASER is to encourage contribution to the cultural environment of a region by fostering interdisciplinary dialogue and opportunities for community building. We want to extend our enormous thanks to the Leonardo Laser team for supporting our application to become the first Australian site for these talks. Both Elizabeth and I have been avid fans, readers, authors and referees for Leonardo for many years, so we're just overjoyed that we get to expand our engagement in this manner with the remarkable institution that is Leonardo. We want to thank the UQ Art Museum for hosting tonight's event. We also thank our respective institutions, the University of Queensland and Southern Cross University, as well as the Australian Research Council for supporting the event. A special thanks to John Edmonds for his incredible organisational work in bringing this event to life. I'd also like to thank ANAT, the Australian Network for Art and Technology, for their promotional support, for their auspicing of a vibrant art science scene in Australia, and for championing the wonderful work of tonight's guests. Which brings me to the end of my spiel, and it's with great pleasure that I'd like to pass over to Elizabeth Stevens to introduce our guests and begin the proceedings. Thank you.
Thank you, Grayson. Well, welcome everyone. What a treat it is to actually be welcoming people to a live event. It's been a long time since we've been able to do that. So it's really a delight to see human faces in a room and not just behind a screen. So welcome to everyone who's joining us here in the room tonight, and welcome to all of our friends and colleagues who are joining us on the stream of this event as well. I'd like to start by acknowledging too that we are meeting on the unceded land of the Yagara and Turrbal people, and to pay my respects to elders past and present and to future generations. And I'd especially like to extend a warm welcome to any Aboriginal and Torres Strait Islander people who are joining us here this evening. You might notice the wonderful image that we're standing in front of here. This is by one of the our wonderful local Indigenous artists here in Queensland, Tony Alberts, as well. So you can continue to enjoy that as we're talking this evening. So when my good friend and colleague Grayson Cooke invited me to join him in launching the first Leonardo Laser Talk series here in Australia, I really jumped at the opportunity to have the occasion to bring together a series of wonderful artists and scientists to talk about their collaborative projects and their issues of shared concern. As some of you will know, this is in fact the subject of my ARC Future Fellowship, which examines collaborations between artists and scientists. And I should say thank you to the ARC as well for supporting this event this evening. So I'm really delighted to have the opportunity to introduce our two speakers this evening, who are both leading and important figures working across the apparent divide between the arts and sciences. And this is reflected in the project that Grayson has already referenced, their ANAT Synapse Award, which they'll be talking about a little bit this evening. And I'm especially excited to introduce our first speaker this evening, who is joining us from Hobart today. So it's a bit exciting to get someone in from out of town to join us at the moment. So let me begin by introducing Svenja Kratz. I first met Svenja Kratz back some time ago when she was doing her PhD in biotechnology and contemporary art at the Queensland University of Technology. And I've watched with great pleasure in the intervening years as she's become such a significant figure as a practitioner in the field of transdisciplinary creative arts practice, particularly working at the intersections between the sciences and arts. Her work includes The Absence of Alice, a series of exhibitions inspired by her engagement with the South 2 bone cancer cell line, and the Human Skin Experience Equivalent Project, a jewellery project involving tissue engineering practices. Svenja has exhibited her work at a range of national and international venues, too many for me to itemise here, and is currently based in Hobart, where she works as a lecturer in interdisciplinary creative practice within the fine arts discipline at the University of Tasmania School of Creative Arts and Media. So Svenja will talk first, and then after Svenja, she will, Dietmar Hutmacher will join her to talk about his part of their collaboration. So I'll introduce Dietmar as well now, and then I will get off the podium. So Dietmar is one of the pioneers and world leaders in the field of biomaterial science and tissue engineering and regenerative medicine. He has a wide range of extremely high impact publications in this field, while also illustrating a real commitment to interdisciplinarity. And I think tonight is an example of that, Dietmar. His research program works across and draws together a wide range of disciplines, including science and engineering, and within this, bioengineering, biomaterial science, computational modelling, chemistry and nanotechnology, as well as the life science disciplines incorporating molecular and cell biology, stem cell research, genomics and bioinformatics, and clinical research, including orthopedics, plastic and reconstructive surgery, and radiology. He's one of the few academics who has successfully translated a tissue engineering research program from fundamental research into routine clinical application. We are so lucky to have these amazing speakers joining us here tonight to talk about their work. So please join me in welcoming to the podium Svenja Kratz.
So thanks so much. Before I begin, I'd also just like to acknowledge the traditional custodians of the land from which I'm presenting today, the Turrbal and Yagara peoples, and pay my respects to their elders past, present and emerging. I'd also just like to take a moment to thank Elizabeth Stevens and Grayson Cooke for inviting me to speak today. As a long-time follower of Leonardo and the associated Laser Talk series, it is a great privilege to be invited as one of the speakers to launch the inaugural Laser Australian series at UQ. It is also wonderful to have the opportunity to discuss collaborative work that I have developed with project partners, including, of course, distinguished Professor Dietmar Hutmacher. And I'll begin my presentation. I'll just see if that will swap over. Excellent. With an introduction to cell and tissue culture to provide some initial context for the talk and the project, alongside some precursor works that give insight into my practice and the foundations of our current project, the Posthuman Genetic Legacies project, which is, as already outlined, part of the 2021 ANAT Synapse program and currently in development across the School of Creative Arts and Media, the Centre for Law and Genetics, and the School of Medicine at UTAS, and of course also the Centre for Regenerative Medicine at QUT with Dietmar. So this project, as you'll come to see, aims to create an alternative genetic legacy via the immortalization of cells from my tissue. And over the next 20 minutes or so, I hope to track some of the key moments that led to the project's conceptualization and will maybe make you think that the project is less weird. All right? So for audience members unfamiliar with my practice, I started my foray into the world of art and science during my PhD, which I undertook, as Elizabeth mentioned, across visual art and biotechnology at QUT, And I was really, really fortunate to have supervisors from both disciplinary terrains. And even though I was an artist, I was actively integrated into the then Tissue Repair and Regeneration group within the Institute of Health and Biomedical Innovation, and trained to actually undertake cell and tissue culture myself. So the first flask of cells that I received were SALS2 cells, where the SALS2 title references their origin as sarcoma osteogenic. So a bone cancer cell line which was established in the 1970s from an 11-year-old girl I called Alice. So these cells, as shown on the slide, are typically grown in a single layer on the base of a tissue culture flask within a liquid nutrient medium. Cell lines like SALS2 cells are considered immortal because, unlike healthy cells that have an inbuilt mechanism to prohibit ongoing replication and thus avoid errors creeping in, which is dubbed the Hayflick limit, they can divide almost indefinitely. As such, they can continue to grow and be split, as you'll sort of see on that slide there, into more and more tissue culture flasks as they fill up the available space, a process called passaging. So this allows vast quantities of cells to be produced, and the total biomass of many common cell lines now vastly outweigh the bodies from which they originated. Indeed, as tissue culture and art pioneer Oren Kratz often points out, the first immortal human cell line, the HeLa cell line, a cervical cancer cell line created in 1951, albeit without the patient's consent, could likely fill this entire area, this entire building, and then some, with an estimated biomass as documented in the book, The Immortal Life of Henrietta Lacks, at 50 million metric tons. I'll just let that sink in for a moment. So the cell line HEC293, while some cells like the HeLa cell, which I've just talked about, are naturally immortal due to the genetic mutation that rendered them cancer cells, healthy cells can also be transformed into immortal cell lines through the exposure to chemicals or the introduction of viral DNA that disrupts replicative senescence or disrupts the stop switch for cell replication. So the cell line HEC293, which you'll see on this next slide, for example, is an artificially immortalized cell line, which was again established in the 1970s, but from the tissue of an aborted fetus. Indeed, the title HEC refers to the cell line's origins, although now somewhat questionably as human embryonic kidney cells. The number 293 documents the number of experiments that were required to immortalize the cells, which was done using sheared adenovirus 5 DNA. To date, HEC293 cells have spawned at least 12 daughter cell lines, which were catalogued by researchers in 2018. So this includes the common HEC293T variant, and here the T signals a further alteration to the cells through the incorporation of the simian virus 40 large T antigen, which basically just refers to a specific protein from an animal virus. And the addition of that particular protein renders the cell line easier to transfect, so to introduce additional genetic material, so RNA or DNA, into the cell. So indeed, during a residency at Symbiotica, which is a leading art science lab at UWA in Perth, I was really easily able to introduce green and red fluorescent proteins, which are derived from marine invertebrates such as jellyfish, into 293T cells. So despite this being a really routine process, what really struck me about working with HEC293T cells was how an organism that never really reached full gestation could nonetheless result in a whole series of unique microorganisms with different traits that are, in a sense, primed for further modification, drawing attention to the ever-present but increasingly porous nature of taxonomic distinctions. In the context of immortal cells, it is also worth noting that despite the implied constancy that the term immortal implies, even without further modification, cells in themselves, the cell lines in themselves are not static, but dynamic entities that are highly responsive to different culture conditions, such as changes in media or temperature. Furthermore, the longer that the cells are cultured, the more they change, becoming arguably ever different from the original donor cells. That is why lower passage number, or cells that have not been doubled and split so many times, are preferred for research, because they have less chance of alteration or mutation, so phenotype or genotype changes, and the cells thus express characteristics that are more in line with the original cell line. As such, to me, cell lines occupy a strange in-between territory, operating as living and evolving remnants of an absent body. They remain intimately connected to the original donor via DNA, yet as cancer or transformed cells separated and living in a laboratory environment, they are fundamentally different from their healthy counterparts and can be regarded, as Oren Kratz and Untitled Chapter from Symbiotica assert, as semi-living entities and a new class of bioengineered life. In returning to creative practice, these reflections were translated into the development of a large series of interrelated works under the collective title The Absence of Alice, that commented on the origins and uncanny nature of both the SARS-2 and also the HEX cell line, but also foregrounded notions of transformation and the potential of biotechnologies to create new forms of life through technological and interspecies couplings. Moving forward, in 2015, I was fortunate to collaborate with Dietmar and his regenerative medicine team on an art science project titled Biosynthetic Systems. Funded by a Creative Sparks grant from the Brisbane City Council, the project aimed to explore the possibilities of biofabrication, so the use of 3D printing technologies for regenerative medical applications such as bone and tissue repair. And as you can see on this slide, the group has a strong track record of translation of lab research to clinical application, such as designing customized biodegradable scaffolds for correcting tissue and bone malformations. So this example here shows a young woman who was fitted with a custom-made scaffold to correct a birth defect that had left her with a sunken chest. The scaffold was implanted along with the patient's own fat cells, and as they proliferate through the structure, the scaffold will eventually dissolve, leaving the patient's own tissue in place. And on this slide, you can see some additional biofabrication examples, including electrospinning, which enables the creation of really fine filaments that look a little bit like spider silk, and of course 3D printing that enables the development of replacement bone structures, but also strength testing for bone scaffolds, and I'm sure Dietmar will provide some more interesting insights. So initially, the project was pitched as an investigation into the ethical implications of developing living products, with the plan to create a sculptural bioreactor to showcase a living custom sculpture with a digital representation of its aliveness via a holographic projection. But as with many collaborative projects, these initial interests shifted in response to the research interests and insights of the larger project team, including Dietmar, of course, artist Bill Hart, and a range of experts from the Centre for Regenerative Medicine, to focus more on the concept of immortality through technological intervention. And this interest was spurred by the recognition that popular film and media representations of tissue engineering often suggest that 3D printed organs and replacement body parts are really, really close to realization, and that soon we will not only be able to repair and reshape the body, but eliminate death altogether through the development of bioengineered or robotic bodies coupled with a form of downloadable consciousness. So the idea of technological immortality is particularly evident in the views of transhumanists such as Ray Kurzweil and philosophically aligned projects such as the 2045 Avatar project initiated by Russian entrepreneur Dmitry Itskov. As you can see on the slide, the 2045 project proposes the development of a series of progressive technological avatars from a robotic body controlled via brain interface to a wholly holographic entity. There are, of course, a range of concerning issues that accompany this proposition, including, again, the positioning of the body as inherently flawed and technology regarded as superior. This is a rather baffling viewpoint when we consider rates of mechanical breakdown and the increasing rapid technological obsolescence we're all experiencing. It also suggests that, in essence, biology can be reduced to binary code, privileging information over materiality, reinforcing the mind and body split, and ignoring the myriad interspecies and embodied connections that make up our physical being. The regenerative medicine team also commented that the regenerative capabilities of the body are still regarded as superior to current technological equivalents, and that many clinical products in development at the centre, such as the earlier example, are designed to harness the healing capacities of the body. To reiterate this point, the team is also working on custom biodegradable breast scaffolds for breast cancer survivors that are designed to dissolve as the patient's own cells permeate the structure, thus ultimately replacing the initial scaffold with human tissue. So through ongoing discussion and experimentation, the Biosynthetic Systems project resulted in the development of a range of creative works and speculative prototypes that enabled us to be playful and test rather than fully resolve our ideas. So works as part of the project included a sculptural biomonument, which featured a mirror hologram of cell growth alongside live 3D printed structures created by mapping the growth and movement of cells on a custom electrospun scaffold. And this work was developed to test the capabilities of the electrospinning system by integrating handwritten text, but also aimed to highlight the dynamic nature of the cells. The exhibition also included a writing machine, which was developed by Bill Hart, that replicated my handwriting and used programmed neural networks to create an ongoing narrative that reflected on what it might be like to be an AI entity and the change from biological to technological. The work was also presented in conjunction with digital video works based on 3D scans of my face that operated as strange and fragmented digital avatars. Creative works were also displayed alongside technologies developed or used by the Centre of Regenerative Medicine, such as a 3D printing demo and series of scaffolds to provide insight into the current developments, but also the limitations of regenerative technologies, and invite viewers to consider the potentials of the Centre's research, but also the philosophical implications of working with living systems and patients, and the potential mergers of artificial life and biotechnology. The monument and writing machine prototypes that were developed initially during this phase were later refined for exhibition as part of the spare parts program at the Science Gallery in London. The writing machine, retitled Ghostwriter, included an expanded text archive, an interactive video avatar, and also a live microphone that would listen to the environment and trigger the machine to write in response to detected words or sounds. The monument was also re-envisioned to comment more directly on the human desire to control the elements and nature in order to find the secret to eternal life. As such, the monument incorporates a range of sigils composed of historical and alchemical symbols, such as the Philosopher's Stone, as well as references to the transhumanist movement. And you'll also see in the centre it features a central biaxial bioreactor showcasing a range of custom scaffolds in the shape of different body parts. Videos in the monument to immortality structure on the exterior also comment on the power of visual culture and influence of the media, science fiction, and popular science imagery on public perceptions of technological progress. The pyramid housing at the top of the monument, for example, features a holographic video that references the concept of the soul or vital force, raising questions about how religious beliefs or spiritual beliefs fit into future projections of technological immortality and replacement bodies. In combination, the two works and different elements aim to really encourage viewers to contemplate their own position regarding engineered eternal life. So more recent work continues this interest in immortality, but with a shift, which I've already alluded to the concept of continuance via the establishment of a genetic legacy. While this focus does build on earlier work, it is also responsive to my situation as a single woman in the later stages of reproductive viability. And over the last few years, I've really started to consider how I might be able to use biotechnologies to establish an alternative biological legacy beyond giving birth to human offspring, such as engineering other organisms such as bacteria, or more interestingly, but with much greater difficulty, tardigrades or hydra, because they are known for their relative indestructibility, and to use them to host fragments of my DNA. And the first work to emerge from this initial proposition was titled Self-Portrait 2, Site of Infection, and it consisted of a wax cast of my bust, rendered in a style reminiscent of classical depictions of female deities that emitted vapor containing my DNA. The work drew on developments in genetic engineering and gene therapy, but also the significant presence of viral DNA, estimated at up to 8% in the human genome from ancestral retroviral infection. Indeed, I propose that rather just engineering bacteria or other organisms to host my DNA, I could engineer viruses to infect and replicate fragments of my genetic code into a whole range of different organisms, including humans. By using a retrovirus and targeting germ cells, so egg cells and sperm cells, it might even be possible for the selected DNA sequences to be then also passed on to the organism's offspring. In this way, I could pass on a portion of my genetic information without having any children of my own, because other people like you and your children, would be able to host and become hosts and continue to perpetuate a tiny portion of me in perpetuity. What an exciting proposition, right? So the work was, as you can imagine, designed to be deliberately provocative and highlight the uncomfortability that many of us experience at the prospect of infection, especially now, and the work was developed pre-COVID, and the idea also of genetic contamination, despite my alignment to the post-human position that we are not so much individuals, but rather multi-species ecologies already comprised of shared and swapped genetic material. So most recently, and as part of our Posthuman Genetic Legacies project, my work has returned to cell and tissue culture, and particularly the prospect of establishing a series of immortalized cells, a bit like the HEC cell variants, from my own body via a range of processes, such as using viral genes to disrupt programmed cell senescence, but also to make the cells available for scientific and artistic use. And cell immortalization is now a relatively common practice, and there are even companies that you can see on the slide that offer an immortalization service, although I'm hoping to do it myself. So in coming up with this idea, is it important to acknowledge that there is an artistic predecessor, the Billy Apple cell line, which was initiated by artist Craig Hilton in 2010. And while the project, The Immortalization of Billy Apple, shares the aim to develop an artist cell line for use across artistic and scientific domains, the project, as you may gather from the registered name, was ultimately about preserving the artist's brand. In my case, the desire to produce my own cell line links to the previous self-portrait project discussed, but ultimately developed from my recent experience of uterine pathology. So in 2019, I was diagnosed with a large fibroid, which is essentially a benign tumorous growth of the uterus that is quite common in women of later reproductive age. And as you can gather from the slide, required surgical removal to alleviate a range of associated symptoms. So essentially, I saw this as an opportunity to not only have tissue to isolate cells and immortalize them from the particular fibroid, but to also use the project as a mechanism to consider how creating alternative offspring from this tumorous mass might offer a sense of empowerment and a space for expressing and rethinking the often unspoken anguish of situational childlessness or potential infertility, which was something that I was really grappling with over a number of years, and particularly during my diagnosis. So prior to having the tumour removed, I developed a series of preliminary works for a solo show titled Morning Story, Expectations, Absences and the Potentials for Self-Persistence, that aim to give some insight into my feelings of grief, reflected in the sculptural works Memento Mori and Coming to Terms with Being Forgotten. Other works addressed problematic constructions of femininity and the legacy of hysteria in the treatment of female reproductive health, evident in the combined Bleeding Table and Clock works, Time is Topological. On this slide, you can see an anatomical model with a snake representing the theory of the wandering womb, the belief that the uterus wanders about the body of a woman like a wild animal. And this condition is the condition that causes major illnesses in women, which was an accepted view documented in early medical texts from ancient Greece, including the Hippocratic corpus. The work SVKRLM, Tumour Baby, in contrast, shifts from a sense of mourning and pain to a more hopeful stance, as it alludes to the establishment of a cell line from the fibroid tissue. By incorporating a flask of fixed cells of mine established from another project and taxidermy butterflies as symbols for transformation, the work makes a direct link back to the work HEC 293T, the transformation of Joni Oliver, which you would have seen in one of the previous slides, signalling also the potential for the fibroid cells to not only establish a single cell line, but spawn a range of daughter cells that open themselves up to further becoming. On the slide, you can also see images of cells successfully isolated from my little tumour baby by project partners Brad Sutherland and Jo Marie Courtney from the School of Medicine. And thanks to ANAT, I will now be able to work with Brad and Jo Marie alongside Jane Nielsen from the Centre for Law and Genetics at UTAS, and of course Dietmar from the Regenerative Medicine Group at QUT on hopefully establishing the second artist cell line for inclusion in the American Type Culture Collection, or ATCC, which is the central global repository for cell lines. But more so, I'm also interested in using the process of establishing the cell line to highlight some of the troubling histories of cell culture and examine current legal and ethical frameworks for the management and use of biological materials across multiple terrains and contexts: creative, clinical, and commercial, including consent, privacy, patent, and IP considerations. But it's also very much a way for me to rethink the boundary of the body and self, and to consider alternative notions of kinship, and also how this might operate in a space that could be regarded as, again, my colleagues Oren Katz and Yonat Zur have observed, as quite exploitative of life and the semi-living. So this begs the question, what are the ethical implications of developing alternative genetic offspring that exist as biological products in a research context? So I will leave it there in the interest of time, and I would just like to take a brief moment to acknowledge the many organisations and people that have supported the projects presented today. And thank you for your attention, and I will pass over to my esteemed colleague.
Welcome. As said by our moderator, it's really great to meet in person again after such a long time. But I also would like to acknowledge the traditional custodians of the land in which we meet today and pay my respect to the elders past and present. And I also extend my respect to the Aboriginal and Torres Strait Islander peoples, which might be here today in person or maybe virtually. So can we have my clicker? I need to use the clicker like a real conference. Okay, so first I have this little disclaimer, which is quite important. I mean, you have heard from Svenja that some of our work has been commercialized and has been translated to the clinic. So yeah, as a scientist and engineer, we work with outlines, as you can see here. So I will talk a little bit about my background, and then about our regenerative medicine projects. And then I will, at the end, spend some time on the collaboration with Svenja, and which I might term the convergence of art, creative industries into regenerative medicine. which I think is a very important part. As said before, my original training is engineering. Then I did an MBA. I worked in industry for 10 years. Then I decided that I don't want to spend the rest of my life in industry. And I decided to do a PhD in Singapore at NUS in the area of tissue engineering. But I was always still very passionate to bring in the creative mind, because I think that's very important. And that's very often not forgotten with the engineers, but especially I think in the area of regenerative medicine, it's very important to get this creative stimulation, which then leads also to the ethical implications Svenja talked a little bit. And I think if you don't have this creative part, it will be very difficult to get to the ethics. So I have to click, yeah. But can you maybe play the clip? So I'm a director also of an ARC PhD training center. So it was great that you acknowledged the ARC, which is a great funding agency. And that's where we do a lot of our work and related to 3D printing and then additive biomanufacturing. So over the years, actually the center is now coming to its end after 5 years. So we had a couple of also events. Yeah, so that's, can you click the other one, please? Yeah, if you go back again, the video should come. Too much technology. Okay. It's, no, but how do I see this one? Do you have to click this? Go down. Down here. This is what they say. Okay. Yeah, it comes. Yeah, so we have been asked to a few times to participate in this TV show, which should normally come with me speaking, but maybe I can just talk to it. So again, this is where I explain that 3D printing really is a major part now in a lot of products you are using, and has come a very long way. And we are also using the 3D printing in the medical field. But as Svenja mentioned, one of the big issues, technically, firstly, is that I have colleagues who talk about that we can print tissues and organs, which is completely wrong. It's a little bit of my passion to really make this clear, because you hear this over and over again. Because the important part is that if you say that you can print tissues and organs, is that you completely neglect the biology. Because what we can do is we can print cells, like the ones Svenja mentioned, which we are isolating, in what we call a bioink, which is a specific biomaterial. So we can mix the cells into this bioink, and then we can print the bioink with the cell. But that is not a tissue, and this is not an organ. These are just cells, which again, and that's where we have the fantastic discussions between the artist and the scientist, is a living object, but just a cell, but it's not a tissue. So it's very limited what the cell can do, right? Because a cell, for example, cannot pump blood, right? For that you need a heart, an organ, which is composed of many cells and so on. So again, this is very important, I think. Then the other aspect, which is also quite important, is this complexity. And this field of tissue engineering and regenerative medicine is now 4 decades old, one can say. And I had colleagues 20 years ago who promised by today we would have the tissue engineered heart, we would have the tissue engineered kidney, we would have the tissue engineered liver, which would be awesome. I would jumping over the roof if this would have happened. But unfortunately, to disappoint you, we are far away from this. We are far, far away from engineering an organ. But again, in the mainstream media, this is still unfortunately very, very often communicated that we can do this. And again, one of the ways I think to communicate this to the society is together to work with artists and make this clear, because if I would make this quite technical, then I think the message would become more complex. So this is already too far. Maybe I'm not going to play this. So I wanted to play you another video, which shows again one of the patients we treated now 2.5 years ago. So our scaffold technology for bone regeneration, where we 3D print scaffolds from biodegradable materials. But this is just the material. And then we bring in the biology by sometimes using cells of the patient, but often growth factors, or by doing specific surgery. And I wanted to show you the clip where, in collaboration with Dr. Michael Wagels, who is a plastic surgeon at the PA, in an interdisciplinary team, we treated a patient who had a bone infection of his lower leg, and the surgeons had to cut out 37 centimeters of his tibia. And the patient was 26 years old, and basically the decision was made that they had to amputate the leg and then put in a prosthesis. So that patient talked to Dr. Wagels, and he said, "Anything you can do for me, doc, I'm going with you." So Michael came to me and our team, and then we discussed if there would be a possibility with our technology to regenerate the bone. That patient today is walking again. About 70% of the bone is regenerated. Actually, he started walking again after 12 months. But it was a very complex process with a lot of engineering, with a lot of biology, with a lot of surgical planning. And that patient also had 2 other operations in between, right? But we saved his leg. So that is a reality. But again, this is not by putting cells into a hydrogel printing and then saying, I have printed a bone. So as I said, this technology has been translated and actually more than 50,000 patients, I think today, received the scaffolds, but not all patients with this major defect, a lot of patients with smaller defects. But we have translated this technology now also to another area, which again, as I mentioned before, to save the leg of a 26-year-old is something specific, which I think is very important for our team to have this in mind, that we really can make a difference for our patient. But an even bigger area is now what we are targeting, and Svenja also mentioned a little bit. So there's this area of breast reconstruction. And here you see an example. These silicone implants are implanted, basically 10 implants around the world per minute, right? So a very large number. And unfortunately, they have a lot of mid and long-term complications, which you see here. There are not many, unfortunately, alternatives. And there is a very strange, I think, communication between the clinical teams, the patients, and then other stakeholders like the insurance companies, like the media in this field, and so on. which obviously has a big psychological impact on female patients. So we are tackling this problem, therefore, not just from trying to develop a technology for regeneration of the tissue, but also to bring in more discussion about this communication and this relationship in this triangle with our behavioral economics team, which you see here. This is one of our publications where we really want to stimulate the discussion about because what we see very often, even so, and Svenja mentioned this also before, there's so much information out, right? So for example, if you go to the Breast Cancer Foundation webpage, there's a lot of information, but the way the information for the patient to digest is very difficult. And then the communication with the surgeon, again, is very difficult, the communication with the family and so on. So we try to bring in, also from this aspect, also some more, first the evidence that this is out there, and secondly, maybe some impact, how this can be changed. So again, what is quite disturbing then is when you read this quote. So this surgeon already said in 1977 that there is a significant problem with his implants, right? This is like 40 years ago. But today, as I said, a lot of these implants are still implanted on a daily basis. This technology has not changed, right? Which is, again, I think, very disturbing. So again, I think we have to find a way to do the communication towards these changes, which again, if we just do the techno talk, is very difficult. Because as I said before, I can produce the best scaffold in the world. If I take this scaffold and I give this to a badly trained surgeon, this scaffold will not work, right? So for example, often what also we see with our technology is that the surgeons have to change their strategy in the preoperative planning, in the surgery itself, and also in the post-surgery treatment. So this is, again, a very interdisciplinary approach. There are a lot of surgeons which are, you can say rightly or not rightly, as we are in other things, so much into their concepts that they are not open to this. So again, one of the questions for us is, how do we break this barrier of this communication? And again, I am very passionate about that. I think that working from the creative side will break up that barrier often, because again, if we do the technology talk, we are not stimulating the right mindset to maybe look into this change. So Svenja mentioned, we have translated now this into the first patient. And again, when you think about this patient, young Caitlin has been living with this for a long, long time. Had, I think, overall like 18 operations, and then decided to go on the journey with this new treatment concept. And here you see also Dr. Michael Vagels, who is a surgeon of this generation, is very, very open to be flexible in his mind to go along with these new technologies and to implement those and so on. And especially also from the psychological side, to really bring this passion in for the patient. Oops. Okay, so now we come to the convergence. So as I said before, I'm quite passionate about this, and actually one of my PhD students is in the audience, and I also often have an artist in residence in my lab. And specifically the students which come with the engineering and material science background. When the artists present, it's quite interesting to see the reflection. But then over time, when you see the discussion and how they work together and so on, this is I think, really rewarding. And unfortunately, this is not really quite embedded into the curricula, right? So if you study at QUT engineering or here at UQ. It's no difference. Both universities or any university in Australia and many other countries are really not teaching this, right? Everybody talks about the interdisciplinarity, but you know, the creative and art side is not something which is bridging over. Likewise, the other way around, I think, you know, from the art side, I see sometimes, I have to admit, projects where artists talk about technology and they have very limited knowledge, which is okay, but are not really willing then also to learn more. And they come up with postulations which they link to the art, where the technology side really doesn't fit in. So I think we really have to collaborate and work together. And this is actually an article related to regenerative medicine, which is addressing this, right? And when we think about regeneration, again, from the technology side, I can make very beautiful scaffolds. You know, I can do a lot of measurements, testing up and down, and so on. But next to, if I give this to a surgeon who is not very well trained, right? So now I have a great scaffold. Now I have a surgeon who is very well trained. But now I have a patient who is super skeptical about what we are doing, right? I think if you would do a clinical trial, and I hope we never do this, to compare patients which are very, very skeptical and patients which are really open and willing, because I think that the patient itself has such a big impact that even a great technology, and actually we can see this with implants like a hip prosthesis or a knee prosthesis or in the cardiovascular field. Again, these are implants which have a very high standard. So there's a large group of patients, for example, who live now with their hip implants for more than 25 years. But there are also a small number of patients where the implants go out after a couple of years. And that's related to also the positivity, maybe you could call it, of the patient in respect and not about the technology itself. So again, in this article, this is raised. I mean, the other things about regeneration, which again, Svenja linked, is rejuvenation. We want to live longer and longer, right? Again, you also all know the statistics when the health minister comes up, and then it is discussed that our life expectancy has been extended in Australia for another 3 years, right? But how many people ask how well, with how much happiness, this extended years have been with the patient, right? And there are some other stunning figures. Again, this comes from health economics. If you think about the last 2 or 3 years of your life, 80 to 90% of the health costs in your lifespan are coming then together, right? So these are all things I think we have to consider. And again, I give you now the figures, but is that really something which sets with you, which stimulates your mindset and so on? I'm not sure. I think more the things maybe Svenja talked about when you go into an exhibition, stimulate maybe more your mindset, that you are more aware about this, and then you think more about also your own health, your own lifetime. And when you then see the doctors for the treatments in the future, that you make decisions which come not just from the technology and again the data, right? And this is related here in this article also. You know, you can go back to the Greek philosophers that they already talked about. Obviously, the combination of the body and the mind. But very often, again, when I go to regenerative medicine conferences, this is not a topic. And I can tell you, if and I think I have never done this, maybe one day I will have the guts to do it, to talk like I talk here. I am sure that a lot of my colleagues would be not interested in the topic. Okay, so again, for myself, I was very inspired by the young lady next to me, because here you see a copy of a dissertation, the front page, because she did the PhD, not with me, but in the institute. I was also with my group, and that's how it all started, this inspiration for both sides. And Svenja already talked a little bit about the exhibitions we did, and they have been, again, very stimulating also for my team, because a lot of team members, as you have heard, have been involved. And again, it was quite interesting to see the change, because at the beginning, they were used more for the technical side. But then as they discussed more and more what Svenja had in mind, you could really see how it was working in them and how their creativity got stimulated in that process. And that's something we want to continue with these new projects. It has been a journey, not just, well, how to put it? I mean, the Australian Research Council is a great funder, but we all know often we miss out on funding, and we are not successful with the grants. So also for us, it was a journey. Overall, I think very successful, but obviously to do the work, we need some financial backing. And it's great that we got this new grant, that we can continue this journey. And I stop here. I hope we have time for questions. I thank you for listening. And if you want to get a little bit more tech information, a mixture of tech information and interdisciplinarity, you can also listen to my TED Talk. Thank you very much.
Yeah, fantastic. So I think we can do questions sitting on our chairs here now. So we have like 25 minutes for questions now, and we will take questions both from the room here and from the Zoom room as well. So Grayson will moderate those too. And is the microphone for the audience?
I think so.
Okay, so we have, oh yeah, maybe we can ask. So with that, then probably only need one. So Johnny, you're going to be our microphone person? Is that, have you just been? Okay, great. Okay, so now, and then so we will have questions for 25 minutes in the room and online. And then we will invite you to join us for a drink and some informal discussion after that. So I'm going to throw it open to the room now. I think I can see everyone. I was hidden behind a pole that I can't see. So who would like to kick us off? Yes, Warwick, is that you waving your hand there? Jump in.
I'm very interested in your idea of immortality and cells. In my particular line, I've kind of thought about that very issue. And I was wondering how you, if you'd like to sort of develop a little, briefly, how you actually understand immortality, because it obviously has particular philosophical and religious connotations, and which don't seem to line up with a materialist science. And yet one feels that there's something like that needed in the thinking that's required in your transhumanist approach. And the thing that I didn't quite get, I might have missed it at the beginning, was that if something is immortal, then obviously then it's not mortal. And mortality relates to death. Something lives and then dies. And so I'm just wondering whether the idea of death is of issue with you in your work, and whether the fact that you're working under the sign of immortality in the proliferation of certain kinds of cells, whether the fact of death that happens, I guess, in cells is something that you're trying to develop to transcend, and if so, if you're trying to transcend it, into immortality from mortality, then in some sense, then, you know, is that genuine immortality, or is it something like quasi-immortality? It's just simply more life.
So thanks for your question, and I guess I'd really like to declare that I'm not a transhumanist at all. I'm actually really, you know, I don't think that this idea of immortality is really useful, and so part of it that I want to use the project for is to actually sort of explore my own relationship to the idea, and particularly, you know, for me, it's been very much an exploration of recognizing my own mortality and kind of going, well, if I don't have children, what am I going to leave behind? And that's been very much this consideration. And so that sort of sparked this idea, well, maybe instead of having children, I could have an alternative legacy. But I also think even in just constructing cell lines, there's a futility in it, because part of me goes, do I really think that the cell lines are going to be really immortal? I don't think so, because I don't think that even if you, and Dietmar would be able to talk more to this, that I think if you actually cultured a cell line for ongoing, which I don't think many people have really done for a really long time in a considered way, but I don't think that the cells would actually prove to be immortal. I think that there is a finitude to all life. And also in one of the works that you would have seen there, the coming to terms with being forgotten is really a way for me to sort of think about, well, the idea is sort of ridiculous, because even this idea of having a portion of DNA that represents me, I think is really silly, because I also kind of go, we're part of a huge continuum that we're connected into deep time. And so we're really just a tiny little part of an ongoing legacy. And so part of me is grappling with this recognition that goes back to more of a post-human view rather than a transhumanist view, that we're all kind of connected into this long history, that we're all part of something. So why is it important to have this immortality? Why is it important? And for me, it's like kind of question, because as much as I recognize that it isn't really important, because I'm already, you know, immortal, if I think of the fact that I'm already connected into this huge history of life that spans beyond myself into other creatures. But at the same time, there's this weird part of me that's really sad at this idea that I don't have anything tangible that I can leave behind. And so I guess that's part of what the project is about grappling with these ideas. But definitely I'm not trying to create some kind of immortal legacy, because I really don't believe that that's possible. So I'm really coming at from, you know, trying to use this as a provocation to really look at what is this desire for immortality? Why is it still so persistent? You know, and how do we actually let go of that? Because I think that it's really problematic also when we start to see ourselves as somehow separate from other organisms, from nature, etc. So I hope that that sort of answers more where I'm coming from.
Dietmar, did you want to speak to that as well?
Well, yeah, with a cell line, you know, again, a cell is just a cell, and if you would culture it endlessly, there, I mean, billions of cells which are dying, right? Svenja showed you, you can order this, right? And then you get 1 million cells into the lab, you do an expansion, but then all the cells die and a few survive and so on. So it's just a technical term, you know, yeah. And again, the philosophical part, I think Svenja explained very well, which is important. But again, there are a lot of scientists which don't think in this direction, and they think, and they make this connection between these immortalities, which is obviously debatable.
Yeah, I think people hear phrases like immortal cell lines, and it's such a resonant, evocative concept that the kind of a general public sort of response to that is to understand it in a very different way than you've just explained, Dietmar.
Exactly, yeah. And maybe also then the other link is more to the stem cell. I think maybe that's also the one when we talk about immortality and regeneration, right? That the stem cell and this cell we all originate from, and if we find out what this stem cell does, then we can recreate humans and so on, right? So I think for me, this is a very simplistic view, again, forgetting this very complexity, which leads to that I'm sitting here, that I'm 57 years old, And over the 5 years, billions of cells have been died and new ones have been produced and so on. And this is beyond any complexity I think we can imagine. And if there are scientists who come, and there are some very smart scientists which puts this in very, very intelligent words to try to explain to you that this is possible, I just like you to be skeptical and learn a little bit more about this complexity.
I'm just going to quickly ask a question from online. This just comes from Alexa, and it's for Svenja. And she asks, how does she see her offspring represented in the future?
That's a question that I just can't answer. And I think that what's really exciting about working with a team like Dietmar is that we sort of come in a lot of the time with a particular proposition. But then the nice thing about having an ANAT grant is that it is open to the collaboration resulting and moving in a really radically different direction. And I think that, so I can't say how I would see it originating. I also kind of go, the cells that were frozen down, there were a few vials that had an infection. And so part of me is also even going, they might not be viable. And so there's always that kind of thing where you just really don't know. And I think, but at least we've sort of got the capacity now to sort of start that exploration and allow for an input from all of the different team members. And we have team members from science, but also from law. And I think that that's going to be really interesting to be able to bring all of that together and to kind of think about, well, where do we want to go with this project? So sorry, I can't give an answer to something there.
And we had a question in the room, Karen, here.
Hello. My question actually follows on a little bit from that. Thank you both for really inspiring and thoughtful talks. So I'd like to bring you back a little bit to the kind of ethical implications of the work you're doing, and especially Svenja, you mentioned very briefly working with the legal, cultural, and social barriers to the kind of research work you do. And especially considering that you've been working on ideas of contagion and ideas of replicating your DNA through viruses. I would be really interested to hear if, I mean, you mentioned that your project started before the COVID period. Has it changed throughout this period? Will you have to find a new way to think this type of regeneration, considering the changes in this area?
I don't think that I would really need to change it. I think it becomes much more visceral for people if you encountered the work now after having COVID, you know, come through. But I mean, there's nothing dangerous about the work as well. And I think it just immediately highlights a lot of these, you know, the fears that we have, particularly with COVID. But then I also kind of go, part of me is curious to see what COVID will bring in the long-term future. Because, you know, I think that we're only privy to, you know, this short time span in our own lives. And I just don't like, part of me is a bit curious of kind of going, well, what particular traits do we inherit from having these infections in the way that you have ancestral infections? And we just don't know what's going to happen. I mean, I would probably be very cautious in the way that I'm couching things. And, you know, even with the exhibition, it was really clear for people that did feel uncomfortable that there was no danger, you know, that we had a risk, we'd done a risk assessment. There was clear outline of what was done to make sure that it was safe. And anyone that's worked in a museum or a gallery environment knows that if there's going to be anything that's, you know, you're going to get sprayed with, it's probably been autoclaved, like sterilized, and it's going to be cleaner than tap water for you to get it into a public space. So I don't think it would really shift too much unless I think that I got feedback from people that they were really distressed by anything that I was presenting. And then it's part of a discussion, because I'm not trying to be, you know, when I say I'm trying to be provocative, yes, I want people to kind of listen up so that they kind of start to ask questions and think. But I also don't want to be causing unnecessary distress to people. And I think that's part of the conversation.
We do have another question from online. This is a question that both of our presenters may be able to reflect on. They're asking about the difference between transhuman and post-human as, for example, something, the difference between a person with a 3D printed arm and a person with a heart transplant. So maybe you could reflect on transhuman, post-human and these different forms of kind of, yes, body printing or medical process.
So I'm happy to talk to, I'll leave Dietmar with the regenerative medicine more question, but I think with the transhumanist versus post-humanist positioning, you know, my viewpoint is that, you know, the transhumanists would be people like I mentioned, you know, Ray Kurzweil, and there's very much this kind of emphasis on human superiority, technological superiority. There's kind of, again, you know, privileging of the human and, you know, looking a lot at technology as being able to afford, you know, this great status where you kind of, you know, evolve beyond human through technology. And I think the post-human position, you know, is really, really different to that. And so I often sort of draw on a lot of feminist post-humanists that are really looking very much at radically kind of rethinking the human and actually looking at more that we're actually connected on a more level, you know, field with other organisms, so that when we're trying to, you know, take the human down from this pedestal and recognize that we're actually all animals, we're part of a lineage, we're part of a kind of system of kinship, we're made up of a range of, you know, interspecies connections, we're connected to nature. So it's a really radically different kind of positioning, but again, there isn't that kind of rejection of technology in that thinking, but rather a recognition that, you know, culture and technology aren't opposites, but that they're really fundamentally interconnected. So I think that would be how I would answer the question of transhumanist versus post-humanist, which are very, very different positionings. And then the other question was around the...
Yeah, so again, we cannot 3D print an arm, right? We can 3D print materials which maybe have some functions, but this would be material, not living matter, right? So you shouldn't compare this to a transplanted heart, right? So a transplanted heart obviously is a heart which you get from another person, and I can see, and again, when you read the reports, obviously there is a big psychological issue for the patients, right? In that sense. But then, again, being more technical, the things we do, like when we regenerate the bone, the scaffold is made from what we call a biodegradable polymer. So it's the same polymer if you would have a kitchen accident and you would have a large wound and you would go to your GP, he would stitch you up. And then he would say, when do I have to see you again that you pull out the stitches? He said, you don't need to come back because the stitches resolve. So we use very similar materials in our scaffolds. So the way it works is that the scaffold is there for a certain period of time, anything between 2 and 4 years, to guide this tissue formation and this remodeling. And then the scaffold degrades and it's then completely gone. Technically, it's going into the Krebs cycle, and I stop here technically. Okay, so it's all gone. So nothing left from what we implanted from the material side. We sometimes also implant the scaffold in combination with cells. And there again, we have now 2 options. Either we can take your cells, so we could go to, for example, to your bone marrow or to your adipose tissue and then isolate cells and then combine them with the scaffold. So this would be your own cells. Having said that, there would be a lot of manipulation in the lab, right? With cell culture media and so on. Or there is something which is called allogenic. So we could take cells from another donor, and that would be then used. But what we also know from today, from these experiments, all the cells we explant, they all die in a certain period of time. Actually, a lot of them die in the first couple of days, right? And then there is this interaction with your own body, and then your own body is doing this replacement, right? So I actually don't see this connection to post or transhuman in that case. Yeah.
Great. Thank you.
I can see we have a question at the back as well.
Thanks for the talk, both of you. I've a bit of questions. So we talk about regenerative medicine and developing people, like more individual level, but we are now facing like climate change and running out of mineral resources. Do you see like opportunities there for more sustainable, way to treat patients in the future? And can we promote this more with biodegradable materials, for example?
No, because, no, because there's also no. I mean, I think maybe what you want to get it is a science. Yes, there is a field of environmental science where biodegradable polymers are used. So from a material science point of view, we use very similar materials. But just to give you one major difference, the workhorse polymer we use name is polycaprolactone. Yeah, it's sorry, I have to be a little bit technical. It's the aliphatic polyester and so on, right? And this polymer is also used in a lot of biodegradable environmental materials now, in cups, in bags, and so on. But the difference is the polymer we use for our implants, 1 kilogram costs between $6,000 to $8,000, right? So you would never make a coffee cup which weighs 5 or 6 grams out of a polymer which costs this money, right? This wouldn't be sustainable. But you can produce this polymer as a non-medical grade material for, we can buy this for $20, right? So there is an overlap of the material science, which I think is important. But in respect to the treatment, no. And one has to admit, that the things we do here at the moment, this is really, really expensive, right? Again, it's very easy for me to say here, oh, my team and I, we worked on this like 3 or 4 of us, it was 3 months, right? If you would at the commercial value of just the manpower we would put in, right? This couldn't be done for every patient at the moment. So, but that's how the cutting edge science works. And so the next step is to break this down, that we are quicker, faster, and so on. But at the moment, this is really a high-tech technology and is only for a limited amount of patients.
Friends, we were almost out of time, so I think we will make this the last question. Here.
Hi. Okay. Oh, it is. Thank you for, gosh, it's such a big topic. We could go on forever, but I was really interested to ask each of you, or both of you, what brought you, or what is it that you like about the other person's discipline? I mean, that's kind of what we were talking about here. Because my little bit of experience of working with scientists as an artist is that scientists don't really get how artists think. And I don't know if it works the other way too, but I was taken by Dieter's comment that his colleagues wouldn't like to really hear him talk about this. And I wasn't sure if it was because they didn't want to hear about the ethics or the creativity, but you had said that you thought the creativity in the arts helped you to translate the science, and that's my question. Yeah. What brought you to embrace transdisciplinarity across such supposedly?
I mean, I started working in art and science because I just really wanted to grow mutants, and I came in to the lab. So I was exactly one of the artists that Dietmar was talking about, with a really limited understanding of biology. And I spoke to my then supervisor, and I was like, I just want to grow a mutant. And she was like, okay, let me come in, have a look at the cells, what we're doing, and the tissue. And it didn't click at that moment that from this little bit of cells on the base of a flask, that it would be really tricky to actually produce my own mutant. Since you can't even create organs yet, you know, that have all of the complexity of living tissue. And so for me, it was really important for me coming in to challenge a lot of my assumptions that I was making about scientists. And I think that it was also really great for the team that I was working with to challenge the assumptions that they were making about artists. And I mean, when I say I was trying to make a mutant, there was more behind it. It wasn't just a frivolous idea. But initially, you know, it took a while. My supervisor at the time, Z Upton, she was really open to it because she really saw the value in being able to introduce someone who had such a really wrong idea of biology to actually integrate them, show them so that you could really understand the other positioning. And that worked really well, vice versa. And it was sort of building trust over time, you know, learning the language so that you have the ability to be able to talk across boundaries. But I also think it is about a rapport. And I was talking to Elizabeth earlier and going, you can't just randomly smush together an artist and a scientist and go forth and make art science work. It doesn't really work like that. There is also a kind of, rapport that you need to have, an openness, as Dietmar was also suggesting, you know, to surgeons. You know, part of me goes, I think you need an openness as an artist to really value not only the scientific insights, but also the creative insights that they can bring to a project, and vice versa. And I think that that's certainly something that I found with Dietmar, is just a willingness to listen, to be open, to have a laugh as well. You know, sometimes where, you know, either one of our suggestions might be really ridiculous, but we kind of can laugh about it. We can hold different viewpoints and allow them to occupy, you know, the same territory without, you know, saying one's right and one's wrong. We might disagree, but that's okay. And it's okay to bring in disagreement into the work as well. And I think that that makes it richer. And again, that's something that, you know, has built up over a number of years as well. So it's not instantaneous. And I think that that's sometimes a bit of misnomer around that you can just do that instantly. You know, particularly in universities, they go, we'll give you money and then in 6 months we want a grant output. And I'm going, it doesn't work like that.
Yeah, wow, not much to add. Maybe the other thing to add is, you know, there needs to be chemistry also, right? And I think that that's what you said at the end, right? Because again, everybody talks about interdisciplinarity and multidisciplinarity. And it's not just between the artist and the scientist, you mentioned the surgeon and so on, right? But we are all humans. We are this complex living system. And again, if there is no chemistry between the team or the one who should collaborate, which, as again Svenja said, means also, you know, there is friction, there is discussion, there is disagreement and so on, right? Then this will not work, and I think that's very important. But then in the bigger picture, I think it's really important to create this environment, right? So, for example, in my lab, right? Yes, it is in a way a top-down, right? So I make the decision to bring in the artist, and then I say, we work now together. But I already communicated with the artist, right? So my students and my postdocs, they are exposed. I mean, I give them also the freedom to go out, but mostly they don't do it, right? But then when the artist is in the team, then things get rolling. So yeah, and coming back to my colleagues, yeah, a lot of them are not inspired by this, which is very strange for me, right? I was always inspired by art, by poetry, and so on. And again, I think this is something also which the bigger picture is missing in the science curriculum, right? I think actually on this note, we go into the wrong direction with the STEM field, right? It's so streamlined now to take the arts completely out, you know, and I think we will regret this in the next 20 years because we will have people which are really in this block thinking and will miss out on a lot what makes up, you know, the Greeks would say a complete human.
Seems like a very inspiring note, Dietmar, to bring the full proceedings of this evening to a close. So I think at this point we will say farewell to our online friends who are joining us. I don't know if there was anything you wanted to say.
No, there are no more questions.
Okay, so we will just say thank you to our online friends who joined us tonight and who shared their questions. And please join me in thanking our two wonderful speakers for this evening. Does not have to come to an end this evening. We'd like to invite you to join us for some informal chat over a nice drink and some canapes in the reception area. So those of you who know the way can lead on, and then we will continue the conversation there. Thank you so much for your questions, everyone. Good work.
Yeah.
Team member.
So done.
So wonderful.